All right. Well, it is 8 o'clock. Can we believe it? Let's see. Are we recording? We are. Okay, great. Day 3. You've made it through the week. This is about what I expected 3 days in from this class to dwindle down to the dedicated, the strong. It'll fill in later. All right. I wanted to let you all know that I did post the videos. So they are up now under these little booklets. So if you click into them, I use this for structure and function, but essentially you can see the videos and you just push to next page. So if you've never used these little books on Moodle, they have all the previous recordings. The embryology video from Tuesday is on my YouTube channel. I just haven't added it to Moodle yet. So I will do that. And then I'll add the little intro-y PowerPoint or whatever from I guess the first hour of the first day. So that I will get up. Today, I'll try to get today's lectures up this evening as well, but Friday at the latest. Then you can look at them again if you want over the weekend. Not that you want to. Anyways, today we have a lot of physiology stuff that we're going to try to get through. We'll see how much we get through. We have physiology spillover on Tuesday after we do our next practice exam next week. But we're going to kind of try to follow this generalized schedule of high yield endocrine, then neuro, then gastro, renal, and cardiopulm. So that will be kind of our order of operation. I put cardiopulm at the end, even though it's pretty high yield, because I'm hoping most of it will be review, since a lot of you had me in cardio, and a lot of the physio high yield stuff is stuff that we already covered there. So that's why I put that at the end today. And we'll start with endocrine. Really, you'll find with endocrine, a lot of it is going to be hormones, anterior pituitary, adrenal glands. So we'll lean into those pieces. There are a few mechanisms that we'll go through like cellularly, but that typically those are lower yields. Oh, and I see people's faces. Hello, people online. I'm gonna close your faces. Okay, great. Sorry. All right. So the PowerPoints I have open is all the endocrine ones. So we'll start with the intro PowerPoint, then we'll go into the hypothalamus and pituitary, then HPA, HPO, thyroid, and then insulin. So we'll kind of go through this order. And we'll see, we may go pretty fast through some of these because there's some duplicate information. So we'll kind of skip through. All right. Great. Endocrinology. The beauty is that you just had this class. So hopefully this is fresh in your brain too. So again, these are just the high yield pieces that I think you should really lean into for physio. A lot of it is going to be hormones, a few like hormone receptor interactions, really leaning into the pituitary gland, hypothalamus, and then the adrenals. So when it comes to our endocrine glands and hormones, starting all the way up top, we have our hypothalamus and all of the hormones that it is releasing. I would know the abbreviation as well as the full name for all of these hormones listed on this page. They will sometimes give you the abbreviation. They will sometimes list out the name in full without the abbreviation. And that can sometimes throw students for a loop. So I think it is important to know TRH is thyrotropin releasing hormone, GHRH, et cetera, et cetera. The anterior pituitary obviously then is going to respond to a lot of the hormones that the hypothalamus is secreting. We will go more in depth on these like reactions and these steps in our next lecture, but just as an overview of where we're going. And at the baseline, you should have a generalized knowledge and understanding of where these hormones are secreted, where they act upon, and then what their final end organ of action is. So be able to follow them, trace them down that whole pathway. When it comes to the posterior pituitary, that's a great test question because again, that's a neuroendocrine organ. And the posterior pituitary is really just a sac. It's a holding place for hormones. It's not actually secreting any hormones itself. It's a storage place. So this is oftentimes a great physiologic area for NPLEX to ask questions. And remember though that antidiuretic hormone, vasopressin, and oxytocin are the two hormones that are stored there. They're produced though in the hypothalamus. I've seen NPLEX use vasopressin instead of ADH. So I would just make sure that you have those two interchanged in your brain. Our thyroid hormones that we want to remember that we know is thyroxine and triiodothyronine. They will often give you the full words. They won't do T4 and T3. So again, this is just remembering, identifying, name recognition. Calcitonin as well, and then adrenal cortex, no cortisol and aldosterone. Your adrenal medulla, that's where we have norepinephrine and epinephrine, your pancreas, insulin, and glucagon, your parathyroid hormone, testes, testosterone, and ovaries, estrogen, and progesterone. This is your whole list. If you're able to know these hormones, know their actions, know where they're secreted, and have a little understanding of regulation, so negative feedback loops, you'll be in good shape for the endocrine section of this exam. I typically find that our endocrine course, our endocrine block, does prepare you pretty well for this test. So now we're going to get into a little bit of cell bio, which has been a while ago. So we're going to talk about some of the structure of these hormones. So the majority of hormones in the body are polypeptides and proteins. Remember, polypeptides that are greater than 100 amino acids are typically called proteins, versus a polypeptide that's less than 100 amino acids is considered a peptide, just a smaller protein. They're synthesized on our rough end of the endoplasmic reticulum, and they are synthesized initially as a prohormone. Those prohormones are transferred to the Golgi apparatus. They're then packaged into secretory vesicles and secreted into either the cytosol or oftentimes into the extracellular fluid, where they then get taken up by the blood. Because the key thing with hormones is hormones travel in the blood throughout systemic circulation. Typically, an increase of cytosolic calcium concentration is a trigger for this secretory movement, this exocytosis from the cell. Calcium is a big trigger for a lot of intracellular activities within the body, whether it's musculoskeletal contraction coupling, action potential generation, neurotransmitter release. We're going to see this pattern over and over today in physiology of calcium playing this role of increased calcium into the cell, either through a voltage-gated channel or calcium release from the sarcoplasmic reticulum within the cell as being a great catalyst or trigger for a lot of these activities to take place. So when in doubt, that's a good one. There can be also, besides increased cytosolic calcium, oftentimes our hormones are responding to an extracellular something that's being bound to the cell, right? Whether that's another hormone, another peptide, or some other signaling molecule, but that extracellular response causing an intracellular action, either through our cyclic AMP mechanism or other secondary mechanisms. Yeah. Is there an example of? Yes. It really depends. I mean, secondary, typically our hormones, so steroid hormones are going to be able to pass through the cell. We're going to look at them more specifically coming up. Any hormone or any non-steroidal hormone or larger protein is not going to be able to go within the cell. So it's going to do one of these secondary messenger pathways. Typically, cyclic AMP is the most common because we're using G proteins, and G protein coupling would be your most commonly tested on and seen secondary messenger system. Yeah, good question. Steroid hormones, though, fat-based hormones are able to pass directly through the cell membrane, and they bind intracellularly, typically on actually the nuclear membrane surrounding the nucleus. And so you'll see those have slightly different action than internally. But we'll look at both of them. But the traditional secondary response would be we have something binding on the outside. We have a G protein then being activated, cyclic AMP, then that's going to lead to increase in protein kinases, and then typically enzyme production by actually modifying genes, increasing proteins, or we can see other gene modifications, shutting off certain genes, turning on certain genes, upregulation, downregulation, those type of things. This isn't a cell class, so those of you who've taken an MCAT, you probably know more cell bio than you need to for this test. But there are a few questions on secondary messenger systems, so understanding that will be helpful. So other hormones, so these were our peptide, our protein hormones that bind on the outside and cause an intracellular response. Our steroid hormones, like I said, are synthesized from cholesterol. They're not stored, so they are created and used when we need them. They're lipid soluble, fat soluble, so they can go through that plasma membrane. And then our amine hormones, these are derived from tyrosine. We see this with our thyroid and our adrenal medullary hormones. Thyroid globulin actually incorporates thyroid hormones in the thyroid gland, and thyroxine binding globulin binds in the plasma. So when in doubt, if your hormone or your peptide or your molecule is a protein, it's going to bind extracellularly and cause a second messenger response. If your hormone is synthesized from cholesterol, if it's a steroid hormone, it's going to pass through the lipid bilayer. It's going to bind intracellularly and cause an intracellular response. If your hormone is a thyroid hormone, it is going to bind also intracellularly. So thyroid hormone is the only one that breaks that lipid cholesterol routine, okay? It's the only one that does different. So we couple it kind of with the steroid hormone response because it can go intracellularly and binds. It does this differently. It actually has some transport mechanisms that's able to get into the cell. And we'll look at the thyroid hormone synthesis here later. Okay. All right. So clearance from blood, you're never going to actually have to calculate clearance, but it's just helpful to kind of think about this process functionally. There's two factors that can increase or decrease concentration of hormones in your blood: the rate of hormones being secreted and the rate of removal, which makes sense when you say it, but then we ask questions and it gets jumbled in your brain. So how fast is the body producing these hormones, or how fast is this body secreting these hormones into your blood if they're already stored? And how fast is your body either using them or breaking them down with an enzyme? The hormones are cleared from plasma in lots of different ways. They could be bound with tissues, destroyed by tissues, excreted by the liver into bile, or excreted by kidneys into urine. So think of either organs or your emunctory organs, your organs of excretion or detoxification, and or your biochemical reactions to break things down, denaturing a protein, utilizing an enzyme, or binding them to tissue and changing said hormone. When it comes to receptors and activation, I've already mentioned this a little bit, but this is a summary slide of what I've been spieling. You can have them either bind on the surface, your proteins, your peptides, etc., on the cell cytoplasm, your steroid hormones, or actually in the cell nucleus, which is your thyroid hormones. Okay, so downregulation of receptors. So again, ways we can control hormone action. We can increase production, we can increase secretion or decrease, right? We can increase, upregulate hormone receptors or downregulate hormone receptors. And so this is one of the ways that the body often controls our response to hormones, especially if a hormone is maybe in high concentration in the body, but we're not seeing a great response from the body. We're likely looking at a receptor issue. Either the hormone's not binding effectively, so decreased affinity, decreased binding capacity, or the actual receptor itself isn't present in enough amount to account for the amount of hormone that's in the blood. So receptor proteins can be inactivated or destroyed even during their function. They can also change day to day, minute to minute, hour to hour, right? Downregulation can occur for many different reasons. Some of the more common ones is inactivation of receptor molecules, inactivation of the intracellular processes, so that second messenger system, or even signaling molecules intracellularly. Sequestration of the receptor in the cell, so maybe that vesicle that created said receptor, which is a protein, is stuck within the cell. It hasn't translocated to the surface yet. Destruction of them via lysosomes or internalized macrophages, those type of things, or even decreased production from a genetic level. Okay. Why are these things important? Because this is a great way for them to ask questions. They'll often ask you when these questions come up, they'll say, is it upregulated or downregulated? Or is it, they'll often do a two-part answer. Is it due to the hormone or the receptor being upregulated or downregulated? And then they'll give you a secondary condition. And so understanding, okay, if I have a lot of hormone and not a lot of action, I'm looking at receptors. Either is there enough receptors or are they not binding to the receptors? Or maybe if my concentration of hormone is low, then I'm looking at some issue with hormone production. It's either hormone not being produced effectively, or is it getting sequestered somewhere? Or is the enzyme too active and breaking it down before it can have its action take place? So thinking about these processes, they can be applied to any organ system that deals with hormones, any of these different endocrine organs. So you don't have to memorize each individual hormone response. You can apply this across the board. All right. Hormone receptor complexes. So this is really going to look now specifically at these different ways that hormones are going to bind to said receptors. So we have two different kind of linked receptors, our ion channel linked or our G protein linked. Ion channel linked receptors we see with our neurotransmitter substances. We're going to look at this more specifically today when we do neurophysiology, and we remind ourselves about action potential generation and all of those good pieces that come with that. Depolarization, repolarization, etc. But all of our neurotransmitter substances that will combine with receptors in the postsynaptic membrane have this ion channel linked receptor process. We'll see that there's often a change in the structure of the receptor that allows for opening and closing to allow for ions to go in the cell or outside the cell, sodium, potassium, calcium, etc. With our G protein linked hormone, this is our classic second messenger system. So the binding externally and then the process internally. And I have a picture that we'll talk through here coming up. You can have G proteins that are also inhibitory. So they are not always stimulatory, but they do amplify whatever reaction they're trying to amplify. So it is going to cause an amplification of the response. That amplification isn't always a stimulatory amplification. There are hormones or substances that can cause an inhibitory effect as well. We have enzyme-linked hormone receptors. These are proteins that pass through the membrane only once. The hormone binding site is extracellularly, and the catalytic enzyme binding site is intracellular. So we see this with tyrosine kinase and a leptin receptor. So we'll have leptin gets bound externally. Tyrosine kinase is active internally inside the cell. It's an enzyme specifically active within. And then we have our intracellular hormone receptors, which are really focusing on activation or deactivation of genes. These are anything that can travel through that lipid bilayer and either activate or bind within the cytosol, or they can even go through the nuclear membrane and through nuclear pores and bind within the nucleus. We see this with our adrenal and gonadal steroid hormones, thyroids, vitamin D and retinoid hormones. They all will bind within the cell to protein receptors. They're lipid soluble. And we're seeing then direct activation or deactivation of specific genes on our genome. Okay. How are we doing so far? I know it's like a cell bio class. I know, it's like all the way back to first term structure and function. Welcome back to my life. All right. Okay, so let's go through a few of these. So this is our adenylene cyclase, our CAMP, cyclic AMP second messenger system. So we'll look at this. This image is not actually showing that at all. I have that. This is actually, I don't even think I have it. Yeah, I don't have it in this PowerPoint. I know, I don't know. It's in another one. We will look at it. So ignore the picture for now. This is thyroid. We'll cover that. But our hormone is binding our receptor. This is our coupling. Our receptor is essentially bound or coupled to a G protein. The G protein's inside the cell. The G protein will become activated if the receptor binds to whatever its specific substance it binds to. And then inside, we'll see adenylene cyclase, which is a membrane-bound enzyme, will convert a small amount of cyclic AMP inside the cell. This will activate any cyclic AMP-dependent protein kinases. Whenever I say activates, I could put phosphorylates in there, adding a phosphate group. And so you'll often see NPLEX refer to it as its phosphorylation as the action, which would be correct. That's the activation step, is often a phosphorylation in this response. Once that protein kinase is phosphorylated or activated, that will lead to your enzyme cascade. And that's going to be specific for whichever hormone or substance bound externally. From there, you'll then typically see upregulation or downregulation of genes, or you'll see increase or decrease production of certain proteins or enzymes. Small amount of hormone, large effect. That's the kicker here. I don't typically see them ask questions that are more in depth and kind of knowing this general process. I think the most common one is the phosphorylation. What biochemical action takes place to activate a second messenger system or a protein kinase or et cetera, et cetera. And it's really that phosphorylation step. That's that activation step. There is another second messenger system that's common, but not as common as G protein. And that's phospholipase C. It's a second messenger system. Essentially, the enzyme catalase break down phospholipids in the cell membrane. It uses the PIP2 second messenger. Inositol triphosphate, IP3, will mobilize calcium ions from the mitochondria and the endoplasmic reticulum. And then diacylglycerol, DAG, activates enzyme protein kinase C. Again, a protein kinase gets activated. So same response at the end. Protein kinases are getting activated, meaning phosphorylated, and then we see our enzyme cascade. It's just not using G proteins. So if you see IP3, if you see DAG, don't let them throw you. It's still a second messenger process. It's still the same kind of process, same action, just using a different second messenger tool. I have not seen them test on knowing which hormones or which proteins have either G protein versus inositol triphosphate responses. So I don't believe that would be important information to put into your brain, which one does which. I do think it's good to know the general process, that these are all second messenger system processes, meaning the hormones are binding externally. This also doesn't just have to be for hormones. This could be for any protein or anything that's binding externally or extracellularly. And then we have our thyroid hormones, which thyroid hormones, they are unique in that they go all the way, they translocate all the way inside of our nuclear membrane in the nucleus where they have their action. So thyroxine, T4, and triiodothyronine, T3, are the two thyroid hormones that are able to get all the way inside the nucleus. They bind to receptor proteins there. Those receptors will become activated once those bind, and then you are going to see controlling function of gene promoter segments. So your promoter genes are going to be upregulated typically. The majority of these intranuclear receptors are going to, they're going to do all the functions of thyroid hormones. So you'll see synthesis of new proteins, which can do all these components, right? Affect many other systems. That's the most generic thing ever, but the thyroid can increase growth, central nervous system development, and obviously cardio and metabolism. Thyroid hormones have large effects on both of those areas. Part of the reason, though, that a little bit of thyroid hormone has a long-lasting effect is because it is responding or reacting directly inside that nucleus. It's binding directly at that genome, and it's affecting those genes specifically. So when you're dealing with a patient who you're managing their thyroid condition, if you adjust their thyroid medicine and their hormones are going up or down, that can greatly affect their cardiovascular system and metabolism for a long period of time, even if you just made a slight change in their medication. And so this is why it's something that we monitor pretty regularly when we initiate a medication for a patient. Once they're stable, then we don't need to. But it can have a great long-lasting effect. Okay. All right. Those are our three main mechanisms. So just to give you some flavor of our endocrine system before we get into the meat. This is our hypothalamus and pituitary gland sections. Now we're going to go into the specific hormones. Any questions about general endocrine processes as we go through? Yes? Did you say that all the PAM and phospholipase are extracellular? Extracellular within secondary messenger systems. Yes, so they're going to have an extracellular binding and they're going to have a secondary messenger system intracellularly. And then is there a differentiation between like which one is the steroid one? Steroid is always intracellular. So neither, steroid hormones would be neither. Correct. Your typical steroid hormones are going to go intracellular and bind in the cytosol, yes, to the receptor, and then they will have phosphorylation to a protein kinase and have the same type of cascade. But that trigger isn't through this kind of second messenger process. Essentially, they cut out the middleman. They can go directly in, bind with the receptor, and then activate whatever protein kinase they need to activate inside the cytosol, inside the cell. Thyroid hormones take it a step further and go all the way into the nucleus. Yes. They don't just like. Correct. Yep, they have to bind to the receptor on the outside. So a ligand receptor binding. So the ligand being hormone or protein or whatever we're talking about, they'll bind to their receptor extracellularly, and then they cause that intracellular response. Yeah, it's because they can't get through the membrane. Yeah, it's a great question. Absolutely. And this is why it's helpful to remind ourselves of these mechanisms, because like I said, they can be applied across the body, across systems. So if you can kind of remember and differentiate, you can answer a lot of these questions that are more cell-based. Great. These questions cross over into sometimes they're biochem, sometimes genetics, and sometimes physio, depending on what they're specifically asking for. So that's where you can see these used. All right. We're going to get into the hypothalamus and pituitary glands. This, like I said, was the high yield stuff. I really would feel comfortable with hypothalamus and pituitary. And then if you do, then adrenal gland would be your next big system. And then maybe thyroid and then pancreas. That would be my order of priority for you. So we'll kind of go the same way today. We already talked about some of the general components of the endocrine system. One component to remember is endocrine and nervous system are friends. So be able to understand how their responses are similar and different. They're both controlling the body, but the nervous system is responding rapidly versus endocrine is slow, right? Nervous system has that like localized neurotransmitter synaptic membrane response, so it's quick. Endocrine has to secrete a hormone through circulation. So the hormone has to travel throughout the whole body in the blood to get to its acting point. So nervous system short duration response, our endocrine system's long, which is great. Nervous system neurotransmitter, endocrine hormones. And then nervous system specific, endocrine system diffuse. So we can see kind of these similarities and differences. I've seen them ask questions about comparing and contrasting these systems before. Also keep in mind an endocrine gland is different than an exocrine gland. This is a great test question. Endocrine producing hormones, and there's no ducts, not ducts uvo, ducts ducts. Okay. All right. Exocrine non-hormonal substances are secreted, sweat, saliva, etc., and they have ducts to carry the secretion to the surface. So the important glands, the pituitary, thyroid, parathyroid, adrenal, and don't forget about your pineal gland, your melatonin producer in the brain. But your hypothalamus is the one neuroendocrine organ, has nervous system and endocrine function. You'll also see some have exocrine and endocrine functions, specifically the pancreas being one of the most commonly discussed exocrine endocrine organs. You'll also have other tissues and organs that aren't primary organs of the endocrine system, but also produce hormones. Our fat cells, our thymus, and even cells in the walls of the intestine, stomach, kidneys, and heart. They all have a hormonal component that they can secrete, but we don't call them primary endocrine organs. They would be secondary endocrine organs because their function is elsewhere. Our primary endocrine organs or glands would be these glands I listed up here, and we would probably include the pancreas and gonads in that discussion as well. Chemical messengers of the endocrine system, hormones we've discussed. Autocrines are things that are going to affect the same cell that secreted them. So if I'm a cell and I secrete something and it's affecting me directly, that's an autocrine response. That's an autocrine substance. It's affecting just me. It's not affecting you. If I'm a paracrine cell from a paracrine substance, that's a local acting chemical that affects cells other than those that secrete them. So if I'm a cell and you're a cell and I secrete something and it doesn't affect me, but it affects you, that's a paracrine substance. And we see autocrine and paracrine substances more discussed in the gut, in the digestive tract, when we're talking about different secretions, but we also have hormones in the digestive tract that we discuss as well. So being able to differentiate between these three words and these three types of things that can be secreted will be helpful. Other hormones we already discussed, amino acid or our protein-based hormones and steroid-based hormones. And then target cells, we already discussed those as well. That's just any cell that has a receptor specific for the hormone that's secreted. That would be a target cell. We discussed already our water and our lipid-soluble hormones, but we now do have our picture of our second messenger system. So if you were waiting for that visual, this is it. This is your G protein coupled receptor. So your second messenger system. You can see here your hormone binds to your receptor. Your receptor is intracellularly bound within the membrane to this G protein in blue. Once the hormone binds here, this becomes active. It's going to translocate and bind to our adenylate cyclase. That's then going to activate cyclic AMP. It's essentially taking ATP and converting it to cyclic AMP, ATP being our energy molecule, which is represented by this little like flash of lightning here. And then your cyclic AMP is what's going to do our protein kinase activation via phosphorylation. Once protein kinase is phosphorylated or activated, that's when all the fun enzyme cascade can take place. Essentially, the action or the intent of the hormone can happen then. We already discussed these. If you needed a reminder, this is our other intra or second messenger system that happens from extracellular binding. The actual second messenger part happens intracellularly. And then our lipid solubles. Okay. Oh, I feel like every year I have to change this. All right. Endocrine gland, not glad. I mean, we're glad, but it's gland stimuli. So there's three different types of stimuli that can affect endocrine glands, humoral, neural, or hormonal. Hormonal is what we've been discussing, hormones stimulating other endocrine organs to release their hormones. The example could be the hypothalamus secreting a hormone affecting the anterior pituitary, and the anterior pituitary secreting a hormone affecting its target endocrine organ, whether it's the thyroid, the adrenal gland, a gonad, etc. So those would be hormonal stimuli. Neural stimuli is using nerve fibers to stimulate hormone release, which can happen. We'll see then a neurotransmitter pathway, a synapse, synapsing on a cell. And when that cell has that synaptic reaction, it then releases a hormone in response, instead of maybe transmitting another nervous system response. And then humoral stimuli. This is changing blood levels of ions and nutrients, and then secreting hormones in response. We see this in the gut and the kidney most commonly, right? We have increased stomach acid because we just ate food, gastrin's released, and the body's like, acidic pH. I need to kind of calm down or stop the system. So then we secrete cholecystokinin to either speed things up or slow things down. Okay. The hypothalamus then. So hypothalamus is connected to our pituitary gland, our hypophysis via the stalk called an infundibulum. Love asking about the infundibulum. It's like on every NPLEX. The pituitary secretes 8 major hormones at least. 2 lobes. The posterior pituitary is a neural tissue. It is not endocrine gland tissue. And an anterior pituitary is glandular tissue. And if you remember from our embryology, they come from 2 different areas, right? Our anterior pituitary, they're both ectoderm, but anterior pituitary is going to come from our oral mucosa, actually, that oral floor, versus our posterior pituitary is coming from the neural tube. It's nervous system tissue. So 2 different developmental places, completely 2 different functions, which is helpful for us to continue to think about. We can see our beautiful anterior and posterior pituitary. This is really going to focus on the posterior pituitary. So this is our hypothalamus, our infundibulum, and then posterior pituitary here. These yellow things are our axons. These are our actual neurons in the hypothalamus with our different dendrites, and that's the cell bodies. And then this is where they synapse directly with blood, right? Because we're going to want to release things into circulation. So neural tissue derived from a downgrowth essentially of the brain. It helps to maintain the neural connection to the hypothalamus via the hypothalamic hypophyseal tract. And that's just what this is called here. This tract from the hypothalamus to the posterior pituitary. The tract arises from neurons in 2 different zones, paraventricular and supraoptic. Again, are these tested on? Probably more than any other neurons that you'll find, but I would say low yield at this point in your study, unless if you're like, this is my jam, nervous system. It runs through the infundibulum and it's going to secrete. So this essentially neural response is going to lead to secretion of oxytocin and ACTH, our diuretic hormone. These hormones are then stored in these axon terminals in the posterior pituitary, and they're released into the blood when the neurons fire. So you could think about them almost as like a neurotransmitter in their release. They're stored in vesicles at the end of the neuron, at the end of the axon here. And when there's an action potential sent down the neuron that then is strong enough to signal that we need those to be released, then the vesicles will actually translocate and release just like any neurotransmitter, but it will release directly into circulation. So this would be an example of a neural response, or if we look back here, neural stimuli. Okay. So oxytocin, really our big action is it's a strong stimulant of uterine contraction. It also acts as a hormonal trigger for milk, for lactation. Both are positive feedback mechanisms. This is a great NPLEX question to ask about positive versus negative feedback. And it acts as a neurotransmitter in the brain. It's going to use our tri-IDO or PIP or PIP2 calcium second messenger system. Which if you think about it acting as a neurotransmitter in the brain or having this nervous response, calcium is a big component that leads to neurotransmitter release. So you can kind of couple those 2 pieces together. Our antidiuretic hormone, or ADH, the hypothalamus does have osmoreceptors that can monitor solute concentrations. So this is an example of humoral release or humoral stimuli. The hypothalamus is scanning our body, scanning our blood for this different solute concentrations. And if it finds that it needs to actually change kind of our diuretic state of the body, then it will secrete or stimulate antidiuretic hormone release. If the concentration is too high, then ADH is secreted. It's going to target kidney tubules to reabsorb more water, keep more water in the body, make the solute concentration lower because we're diluting it by keeping more water in. Okay. The release can also be triggered by pain, low blood pressure, and different drugs. Alcohol and diuretics inhibit it. High concentrations can cause vasoconstriction, so it can also be called vasopressin. Press thin. There we go. Okay, these are your 2 posterior pituitary ones. If you got these, you're good. This is also how we could relate our endocrine gland from our inner nervous system to the kidney, right, and to the heart. So we can start to see these interconnections here. Now our anterior lobe. This is glandular tissue. It's oral mucosa outpocking. That's how it was embryologically derived. It's vascularly connected to the hypothalamus via the hypophyseal portal system, which has two plexes and a vein. So the primary capillary plexus, the secondary capillary plexus, and the hypophyseal portal vein. So it's essentially the communicating system between the two. The hypothalamus will secrete releasing and inhibiting hormones to the anterior pituitary to regulate the anterior pituitary's secretion. So again, neuroendocrine process of our hypothalamus. Anterior pituitary, though, is just endocrine, no neural component. Every hormone that's secreted by the anterior pituitary, all 6 hormones are peptide hormones. All but growth hormone activate target cells via our CAMP second messenger system. So all of these are going to be our cyclic AMP, our G protein second messenger process, except for growth hormone. All but 2 are tropic hormones, tropins. They regulate the secretion of other hormones. So essentially they're hormones regulating the secretion of other hormones. They're like the go-between. We could think about it as TSH, right? TSH is regulating what hormones? T4 and T3 in the thyroid, right? That's its job. It's thyroid stimulating hormone. It's telling the thyroid, hey, secrete more active hormone, please. Versus prolactin, prolactin has its direct action from release. So prolactin is one, it's not going out and it's not causing another hormone to be secreted necessarily. It's just saying, this is my action, increase in lactation. Same with growth hormone. Growth hormone it can cause the action of other things to take place, but it's an actual hormone that once released, it's in its active form. So they are non-tropic hormones. So starting with growth hormone from the anterior pituitary, it's also called somatotropin because it's produced by somatotropic cells. So you could see them called growth hormone somatotropin, and I've seen that used both. It will have actions on your metabolism, and it'll also have indirect growth promoting actions. So on metabolism, you'll have some glucose sparing actions. It'll decrease the rate of cellular glucose uptake. It'll help to essentially be anti-insulin effects. So if you could think about glucose sparing, that means less glucose going to the cells. So what will happen with blood sugar? It's going to increase. You're going to see hyperglycemia, right? So if you had too much, if you had inappropriate amount of growth hormone, you could see hyperglycemia because you have glucose that's not getting taken into cells. Instead, it's being left into the blood. So with hyperglycemia, then what could happen with urine? What could you see? I heard someone say it. Glucosuria, absolutely. So we can kind of take a couple steps and walk through like, okay, well, what would happen then metabolically? What would the concentrations of our blood be? What would be happening in our kidney, et cetera, et cetera. And that's how they'll ask questions typically related to growth hormone, is they're going to connect it to metabolism. They might connect it to some biochemistry, and they'll definitely probably connect it to the kidney. I saw a hand. Yes. It can. So you could see, or at the very minimum, over time, the body having a harder time releasing insulin, right? Or insulin resistance. You can see issues along that road. Yeah, good question. And you can see that in some of the pathologies where you see inappropriate secretion of growth hormone. Great. Other pieces, it will trigger your liver to break down glycogen into glucose. Your glycogen stores, your glucose stores are now being broken down. So less storage glycogen in your cells. You're actually then going to have more glucose in your blood, right? So more things that cause hyperglycemia. And you also see increasing blood levels of fatty acids for use as fuel and encouraging cellular protein synthesis. So it's not all bad, right? So it's not just like everything's dysfunctional. In fact, growth hormone's really good if we need sugar fast, right? Or if our brain needs sugar. It's an important thing to help us have that energy available. So it's not a damaging thing. That's why we have it. But when it goes wrong, that's when we can start to think about pathologies and connections there. Some indirect actions on growth, it's going to trigger liver, skeletal muscle, and bone to produce IGFs, insulin-like growth factors. And this is often something we test in patients where we think that there's maybe an adenocarcinoma or a pituitary tumor that's secreting growth hormone. IGFs can stimulate cellular uptake of nutrients to get DNA, proteins needed for cell division. It also can help with collagen and bone deposition to actually essentially increase our bony matrix. And it will stimulate the majority of cells to enlarge and divide, but the most common targets are bone and skeletal muscle, which is why you can see conditions like gigantism seen here, or acromegaly. Yes. You could, because you're breaking down glycogen and you're building. Yeah, I would say it's appropriate. The question was, could you say that growth hormone is both catabolic and anabolic? And I would say that would be correct by the definition. Awesome. Any other questions about growth hormone? Great. So thyroid stimulating hormone, TSH, it's a tropic hormone. It's also called thyrotropin. So you'll see them use thyrotropin most of the time, produced by thyrotropic cells. So when in doubt, if they ask you about a cell type, choose the closest one to its actual name. It's probably correct. It's going to give us normal development and activation of the thyroid. Its release is triggered by thyrotropin releasing hormone, or TRH, from the hypothalamus. So remembering TRH stimulates TSH, which stimulates T4, which gets converted into T3, right? That process. It's inhibited by rising blood levels of thyroid hormones. So that's a negative feedback mechanism in a normal healthy tissue. And it's also can be inhibited by growth hormone inhibiting hormone, GHIH. GHIH is one of the hormones that's produced by the hypothalamus to control growth hormone release. Our adrenocorticotropic hormone, ACTH. Try to say that one 3 times fast. Remember the first time I took I think it was NPLEX II maybe. My math NPLEX 1, I don't remember. But I saw the word and it took me like it felt like forever. It was probably 5 seconds to put that into ACTH. So just again, remember, go with the whole word. And then if you are given the acronym, that's even better. It also is often called corticotropin. It's secreted by corticotropic cells. The precursor is pro-opiomelanocortin. The only reason this is important is because, well, let's see if you remember. Why is that important to know? Releases other things when broken down, like what? What other organ system can it have an effect on? The skin and melanin, yeah, melanin production and melanocytes. So that's why that's the only reason I put this here, because it's a good biochemistry question, and it's also a good integumentary tie-in for the skin. Is the precursor to corticotropin, or ACTH, is pro-opiomelanocortin. And if you break this apart, you'll get your corticotropin, so ACTH, as well as melanin or melanocytes, essentially. So be able to produce melanin. The regulation of ACTH release is by CRH, that's secreted by your hypothalamus. It's a daily rhythm. So kind of like cortisol has a typical rhythm, so does ACTH. It's higher in the morning. And there's also internal and external factors that can affect it, like low blood sugar, stress, and hot temperatures, so increased temp. So thermoregulation will come into play in regards to ACTH release. Okay. Gonadotropins, FSH, bless you, and LH. So follicle stimulating hormone and luteinizing hormone are secreted by our gonadotropic cells in the anterior pituitary. FSH is going to stimulate the production of gametes, egg and sperm. LH produces or promotes the production of gonadal hormones. So in females, LH is going to help mature follicles of the egg, trigger ovulation, release estrogen, release progesterone. In males, LH will help with testosterone production. They are both absent from blood in pre-puberty boys and girls. So that's a great NPLEX question. Prior to puberty, what anterior pituitary hormones are absent in the blood, or not secreted in the blood, or secreted in low amounts? FSH and LH would be your answer. And then the regulation is by gonadotropin releasing hormone, GnRH, which is released during and after puberty. It has a negative feedback loop suppression. Prolactin is secreted by prolactin cells. It stimulates milk production in females. We don't have a great role in males yet understood. There's some roles that we've discussed, but the main action that you would want to know for the NPLEX would be lactation. It's regulated by prolactin inhibiting hormone, PIH, which is the same thing as dopamine. So dopamine is going to regulate prolactin release. PIH is dopamine. They equal the exact same thing. There's just two different names because we call dopamine used as a neurotransmitter. PIH, prolactin inhibiting hormone, we only call dopamine that when we're specifically talking about its control on prolactin in this specific space, in the anterior pituitary hypothalamus. PIH will prevent release of prolactin. This is why we don't have inappropriate prolactin secretion all the time. And so when you see decreased levels of PIH, decreased levels of dopamine, you're going to have, you're going to actually be able to lactate. Increased estrogen levels will stimulate prolactin, which is why breast swelling and tenderness during the menstrual cycle can happen. And blood levels will rise with prolactin towards the end of pregnancy. Suckling stimulates prolactin release and promotes continued milk production. And oxytocin helps too, so they work together in tandem. Okay. I think that's the biggest one is prolactin inhibiting hormone. They'll ask you like what neurotransmitter is most similar or most alike. And literally they're just wanting to see that you know that dopamine is PIH. This is your summary. If you're like me and you like easy charts, this slide's for you. It has your posterior and your anterior hormones, regulation of release, target organ, and what happens if it's hypo or hyper secreted. This would be probably where I would study from. All right. 2 down. How are we doing? We're going to look at our HPA axis, and then we'll probably break. HPO, what does it mean? Hypothalamic, pituitary, adrenal, and ovarian, right? So we're connecting those two. You could put like HPG, right? Gonadal could be there too. We also could throw in kidney because really it's all of them interconnected. My main goal for us is we're going to look at the adrenal gland, and then we're going to look at the big kind of like pathway of synthesis for these hormones. And I'll show you the enzymes I really would want you to remember and to know. So ZG, ZF, and ZR, I'm going to abbreviate them throughout. So ZG is your glomerulosa, F is your fasciculata, and reticularis is your ZR. Remember, when we're looking at our adrenal gland, we're looking at the cortex, not the medulla, the cortex. We're going to go from out to in, GFR. Okay, so glomerulosa, fasciculata, reticularis, from outside to inside. And so you can see here, this is going to show you ZG, ZF, ZF. This is showing you where these things are taking place, in what area of the actual adrenal gland. So cholesterol is going to be our starting backbone for all these adrenal steroids. Pregnenolone is going to be your next step, your next precursor present. That's a great question. If they say, what is the precursor for all steroid hormones? And cholesterol is not on there, but pregnenolone is. That's your next best answer. If cholesterol is listed, cholesterol would be your best answer. If they give you both, I would choose cholesterol because cholesterol is the precursor, the backbone, but pregnenolone is that first step in the pathway. Then you're going to move to 17-hydroxypregnenolone or progesterone in this kind of pathway. 17-alpha hydroxylase is one of the most commonly tested about enzymes. And I have it listed here because it's located and affects 2 different processes. It affects pregnenolone to 17-hydroxypregnenolone, and it affects progesterone to 17-hydroxyprogesterone. There's a pattern here, right? Whatever the precursor was, it's essentially adding a hydroxyl group at the 17th space, right? So 17-hydroxy, that's what 17-alpha hydrolase does. And it's going to add that hydroxy group, that OH group, at the 17th carbon. Okay, so that's what we're looking at here for these 2 enzymes. So it can affect pregnenolone and progesterone. So that's why I think it's helpful to know. We'll look at the full pathway and then we'll find those 2 spots here in a moment. You can see that progesterone and pregnenolone are both in the zona glomerulosa versus pregnenolone, once they're hydroxylated, essentially, they're in the fasciculata region. Okay, so if you have a deficiency of 17-alpha hydroxylase, it will stop the conversion of pregnenolone or progesterone to their 17-hydroxylated form. Cholesterol, pregnenolone, progesterone, and downstream will be affected. It will increase aldosterone because it's going to shunt stuff to that other pathway. Because we still have cholesterol that's being used. We still have cholesterol that's in our body. And so we're still going to create some hormones. if we've blocked two pathways, that has to go somewhere, right? If something's blocked and can't move forward, we're going to have a buildup of those products, of those end products. And so then we're going to start shunting things down a different pathway. And you can see an increase in aldosterone with both of these cases. All of them are occurring within the zona glomerulosa. Therefore, you're going to see an increase in mineralocorticoids. So that's what's produced in that zona glomerulosa space. Cortisol and sex hormones are both decreased in deficiency of 17-alpha hydroxylase. Increased aldosterone gives you increased blood pressure, decreased potassium due to effects on the kidney that aldosterone has. And so you'll see things like atypical genitalia, undescended testes, lack of secondary sexual development. I saw a hand in the back first and then a hand here. Is it still a hand? You could. I would say that's not the hallmark of this, but you could, right? Yeah. Yes, I'm going to get to the picture. And I think let's look at this, and I think this will help. Okay. So if we look at kind of these pathways here, this is breaking it down to zona glomerulosa, zona fasciculata, and zona reticularis. And recognize that these are all kind of interwoven on each other. I have them pulled apart here just so it's ease of seeing what's happening. Aldosterone is here, so zona glomerulosa. And then we have our like zona reticularis, which has our DHA. This is where our sex hormones would be. And then cortisol, our steroid hormones, or our stress hormones. So if we start cortisol and pregnenolone, all the same. Let me get my little handy dandy pen. Can we see that? Great. Okay, so we start at the top, all the same with cholesterol and pregnenolone here. And then we are essentially blocking this conversion. So we're blocking the conversion here. So we've stopped this process. And then we're blocking this conversion here. So we've stopped this process. On this side, blocking this conversion here, because this is the same conversion, pregnenolone to 17-hydroxypregnenolone. So those are the same. We're just showing kind of the different pathways you could follow. So we've blocked every pathway downward except for in the zona glomerulosa. This is the only one that doesn't have this 17-alpha hydroxylase that's affecting its direct pathway, because this is a different enzyme. This is CYP17. This is CYP21A2. So this is a different enzyme used in this pathway. So we're able to move forward to get to aldosterone. So all of our pregnenolone is going to be shunted this direction and then go down this pathway. Does that help? Okay. Yeah. That's what I do when I'm struggling with these processes. This is an okay picture. The better one is when they're all interconnected. And I have a couple of those posted in previous PowerPoints, and I can post a picture too. But I think USMLE has an okay one. I don't love it. So I'll post a picture that's better. But essentially you're figuring out whatever enzymes blocked, what pathways could then the precursors be shunted to. And then that's what you're going to have a buildup of in the body. And then whatever that buildup is, then you can say, okay, if I have excess aldosterone, what's going to happen downstream? If I have excess cortisol, what's going to happen downstream, et cetera, et cetera. Yes. Yeah, sorry, this is not appropriate. So I just crossed that out and it's not supposed to be crossed out. So you can ignore my little scribble here. Yes, absolutely. Good question. Is that the same question here? Alpha hydroxylase. Yeah. 20, this is 21. This is correct. I just got block happy and started blocking everything. I get so excited when I get to draw. So technically, could this pathway go down and go through a seek pathway? Sure, if there are some seek pathways around, but you'll see, and this is why I don't love this picture as much, because it makes it seem like it has this other option. This other option isn't quite as direct as this picture makes it seem. Awesome. All right. All right, I'm going to go back to where we were. So our 17-alpha hydroxylase. So the big piece here is aldosterone. It's going to be our increased substance. It's going to be increased compared to all the others. Can you have a partial deficiency? Sure. So you could have a full deficiency where you have none, or you have partial where you have some. Either way, you're still going to see an increase in aldosterone. It's just going to be the severity of the symptoms. But the big kicker is lack of secondary sexual development here. Now, adrenal steroids. So we have progesterone. So we'd start from the top. We have progesterone, the 21 hydroxylase. So that's different than the 17 alpha hydroxylase. So progesterone, 21-hydroxylase-2-deoxycorticosterone, which uses an 11B hydroxylase to corticosterone to aldosterone. So progesterone can actually get us down to aldosterone. We'll see they all have interconnected pieces of the puzzle. I think, did we get down? So we going, oh, this is cortisol now. Yeah, I was like, where am I going? The other one here, so synthesis again of adrenal steroids to get us down to cortisol. We have 17-hydroxypregnenolone, 2-hydroxyprogesterone. There's again the same enzyme, 21-hydroxylase to get us deoxycortisol, 11-beta hydroxylase to get us to cortisol. So there's a lot of crossover. There's multiple places where you use the same enzyme, which is beautiful and confusing. That's why it's such a good test question. So what happens if you have a deficiency in this guy? 21-hydroxylase. So a 21-hydroxylase deficiency is going to stop the conversion of progesterone to 11-deoxycorticosterone. It's going to also stop the conversion of 17-hydroxyprogesterone to 11-deoxycortisol. So we'll see cholesterol normally goes to pregnenolone and progesterone, 17-hydroxypregnenolone and progesterone, DHA and androstenedione, and then we'll see sex hormones increase. This will all occur within the zona reticularis. So you'll see an increase in androgens. You'll see decrease in glucocorticoids and mineralocorticoids. Because again, we're blocking this conversion and we're blocking this conversion. So a decrease in aldosterone and a decrease in essentially long-term cortisol. And then the only one that you have is sex hormones to increase here. What you'll see is blood pressure will go down, potassium will go up. The opposite effects of blood pressure and potassium as 17-hydroxylase. So you could think of 21-hydroxylase as having the opposite blood pressure and the opposite potassium effects as 17-alpha. And your clinical characteristics, most common presentation will be salt wasting. You could see some issues of excess androgen secretion, but that's not as big of a presentation as like the salt wasting problem. That will be the main thing in the room that you'll see. And the last one I will talk about, I believe, the last one I'll talk about, because this is the most common deficiencies that you'll see tested on your NPLEX in regards to enzymes. You'll probably see all three of these, or at least a couple of them, is your 11-beta hydroxylase that stops the conversion here. Let me get my all erase so it's less confusing. This is going to stop your conversion here because we're affecting this enzyme, and it's going to stop. I look like a mad person when I have all my crazy drawings. There we go. Okay. Stop my conversion here. Okay. So we stop those conversions. We're also going to see sex hormones increase. These are also all occurring within the zona reticularis. Therefore, we'll see increased androgens, cortisol and aldosterone are decreased as well. But blood pressure in this instance increases instead of decreases because of increased 11-deoxycorticosterone, because we went down one additional pathway, right? First time we weren't able to get from here to here. This time we were able to get to here, but we couldn't convert it into corticosterone or aldosterone. So we're stuck at this deoxycorticosterone, which actually will cause an increase in blood pressure. Blood pressure will increase. We'll see lowered potassium, less renin due to increased blood pressure. And your characteristic that you'll see in these patients is severe hypertension in childhood. So typically, not as an infant, because we won't often check blood pressure that young, but first time a blood pressure check in a peds, if their blood pressure is inappropriately elevated, this would be something I'd be concerned about. Severe hypertension, malignant hypertension as a child with normal, typically body habitus. Okay, so knowing these three pathways will be helpful. These three enzyme deficiencies, they're often asked. This is your adrenal steroid processes. You see this is going down to DHEAS, and then this is taking you down to testosterone, estrone, and estradiol. And again, there's lots of sequence pathways. You can go from testosterone to estradiol, you can go to estrone to estradiol. So there's lots of different ways that you can get to the same hormone and response. So if you have an enzyme deficiency, you can still produce some. Oftentimes, you're just not going to have as much. This, like I said, just separates it into glomerulosa fasciculata and reticularis. Salt, sweet, sex, if we want to remember. Salt, aldosterone, sweet, cortisol, and then sex, your androgen hormones. All right. Woo. Okay, that was great. So we're gonna take a break. We've been working. So we'll come back at 10 after, and then we're gonna move into some neurologic discussions. We'll start talking about neurotransmitters. Okay.